Chapter 15, Blood Flow and the Control of Blood Pressure. So in this chapter, we'll talk about our different blood vessels, and then we'll discuss blood pressure, as well as our different blood vessel functions. So resistance of our arterioles, how blood is distributed throughout our body, what happens at the capillaries, what the role of the lymphatic system is in relation to our circulatory system. We'll then discuss how to regulate blood pressure, and then we'll end with a small discussion on cardiovascular disease. So here is a way to look at our cardiovascular system. So here we're going to start from venous return. So we have our vena cava bringing deoxygenated blood into our right atrium. Blood will then travel through the tricuspid valve into the right ventricle. It will then leave the pulmonary valve into the pulmonary artery to the lungs, where the blood is going to be reoxygenated. The oxygen-rich blood will return to the left side of the heart through the pulmonary veins into the left atrium, through the mitral valve or bicuspid valve to the left ventricle, out the aortic valve into the aorta. After the aorta, we have our arteries, and our arteries are unique because they are elastic. So when the left ventricle contracts and pushes blood out, the arteries will expand. And elastic things, after you expand them or stretch them, they will recoil. And so that's going to keep our blood flowing. After our arteries, we have our arterioles. And our arterioles are unique because they can vasoconstrict and vasodilate. So this is where we're going to alter our blood vessel diameter or our blood vessel radius. After our arterioles, we have our capillaries. Capillaries are unique because this is where our exchange takes place. After our capillaries, we have our venules, and then after venules are our veins. And the veins are unique for several reasons. It is expandable. It also has limited contractility, so we can vasoconstrict here. And then it has valves, and these valves prevent backflow of blood. So it ensures that blood flows in one direction. And depending on the purpose of the artery, we'll find different tissues around it. Purpose of our blood vessels, we'll find different types of tissue. For example, our arterioles are going to be the ones that vasoconstrict and vasodilate. So they're going to be surrounded by a layer of endothelial cells and then smooth muscle for the contraction. Capillaries, where exchange takes place, we want the exchange to be easy, so it's just a single layer of endothelial cells. Arteries, remember these are the elastic ones, so we have a lot of elastic tissue here. And then the veins do a little bit of everything, so we have a little bit of everything. The process of angiogenesis is to make new blood vessels. This is necessary for normal growth and development, but it's also necessary for things like wound healing or uterine lining growth. Also, if we have coronary heart disease, we can grow new blood vessels to supplement flow of partially blocked arteries. So let's take a look at our arteries. And remember, our arteries are unique because they are elastic, which means after you stretch them, they will recoil. And that recoil is important because it's going to maintain the pressure for blood to flow when the heart is in diastole. Now remember, when our heart is contracting, and by heart, this is going to be your left ventricle. When our left ventricle contracts and increases pressure, all of that pressure is going to push blood out the aortic valve into the aorta and into the arteries. And your arteries will expand as it fills up with blood. When your ventricles relax, and when it relaxes, that aortic valve shuts. The reason blood is able to continue to flow is because this recoil of our elastic arteries, when our heart is in diastole, maintains enough pressure to keep blood constantly flowing. So that recoil is important. And the ability to stretch is important. And as we lose the ability to stretch, and as our arteries become stiffer, that's how you get high blood pressure. Okay, so let's review blood flow. So blood flow is directly proportional to the change in pressure, and it's inversely proportional to resistance. So blood, remember, will always flow from an area of high pressure to an area of low pressure, and it flows down the pressure gradient. If there's no difference in pressure, blood will not flow. And blood flow is opposed by resistance. And resistance is determined by things like the radius of your blood vessel, the viscosity of your blood, and the length of the entire system. Those are all reviewed from Lab Physio EX. Okay, So what determines your blood pressure? So if we take a look at our mean arterial blood pressure, it's determined by several factors. The first one is blood volume. So the more blood we have in our circulatory system, especially in the arterial circulation, the higher the blood pressure. So what determines blood volume? Well, our fluid intake certainly influences it. The more fluid we drink, that water will become part of the plasma of our blood, so our blood volume increases, which means our blood pressure increases. When we can also lose fluids, and we can lose it passively. We can also lose it through the kidneys, obviously creating urine. So blood volume is quite important. The other is the effectiveness of the heart as a pump, meaning our cardiac output. Remember, cardiac output is the amount of blood pumped out of the heart per minute. So the more blood we're pumping out into our arterial circulation, the more blood we have, so the higher the blood pressure. And cardiac output, we know, is heart rate times stroke volume. So both of those will determine blood pressure. For example, if our heart increases contractility, it's going to increase stroke volume, which means cardiac output will increase and blood pressure will increase. If we decrease heart rate, cardiac output would decrease and blood pressure would decrease. Another thing that influences blood pressure is the resistance of the system to blood flow. And that's determined by whether or not our arterioles are vasoconstricted or dilated. So if we vasoconstrict, blood pressure increases. If we vasodilate, blood pressure decreases. And then finally, we have the relative distribution of blood between the arterial and venous blood vessels. And that's going to be determined by the diameter of the veins. So our veins are unique. It can expand and it has valves, so it can actually hold on to some blood. We can kind of almost store blood in the veins. If more of our blood is being stored in the veins, then that means there's less in the arterial circulation, so blood pressure drops. If our veins are contracting and our skeletal muscles are contracting and squeezing our veins, it's going to push more blood back to the heart, which means the heart is then going to pump it out into the arterial circulation, so blood pressure would increase. So let's take a look at blood pressure and how we can regulate it and help restore it back to homeostasis. So when we increase blood volume, let's say we're drinking a lot of water, that's going to cause an increase in blood pressure. So when our baroreceptors, these are the receptors that sense changes in pressure, sense this rise in blood pressure, it will respond accordingly to try to lower blood pressure. So what can you do? So we have a very quick response by the cardiovascular system. So if we want to lower blood pressure, we will cause vasodilation of our blood vessels and will decrease our cardiac output. For example, this is done by activating the parasympathetic and inhibiting the sympathetic. So that will cause vasodilation, that will lower heart rate, that will decrease contractility, and all of that will contribute to lower blood pressure. On the other hand, we have a renal response that's a little bit slower. So the kidneys will respond by trying to get rid of the excess fluid that we brought in. And so creating urine is slightly a slower process, obviously, than just quickly activating a neuron and changing heart rate. But we will be able to then get rid of the excess fluid that came in to, again, help regulate blood pressure. So to summarize, resistance to blood flow is directly proportional to length and to viscosity, and inversely proportional to the fourth power of the radius, which basically means, yes, length, viscosity, and radius can all influence resistance and as a result blood flow, but radius will have the greatest impact. So small changes in radius have large effects on blood flow. And that's good because it's easy to change the radius of our blood vessels. We just vasoconstrict or dilate. That resistance in our arterioles can be influenced by the sympathetic nervous system. We can also have local control as well as hormones playing a role. And if we think back to how smooth muscle is activated, remember smooth muscles are the muscles that are surrounding our blood vessels. We have myogenic regulation. So smooth muscles, remember, can contract all by themselves. So our blood vessels can vasoconstrict as required. We also have paracrines. Paracrines are chemicals that are released by one cell that acts on another. So in this case, the vascular endothelium can secrete, for example, nitric oxide, and that will act as a vasodilator. And then sympathetic control. So the sympathetic nervous system will cause vasoconstriction of our blood vessels. So here is that sympathetic causing vasoconstriction. So here we have our sympathetic neuron. It releases the neurotransmitter norepinephrine, and it binds to adrenergic receptors on the blood vessel. In this case, alpha adrenergic. And what you're seeing here is when we want to vasoconstrict this blood vessel, the way we would do that is we would increase the stimulus frequency of our sympathetic neuron, which means we would release a lot of norepinephrine, and that will cause this blood vessel to vasoconstrict. When we want the same blood vessel to vasodilate, what we'll do is it's not the parasympathetic that does that. Actually, what we'll do is we'll essentially turn off the sympathetic neuron. And so the action potentials travel few and far between. And as a result, we don't really release a lot of neurotransmitters. And as a result, this blood vessel will vasodilate. This is an example of tonic control. And it's called tonic because the same neuron is able to do both. We'll vasoconstrict when we turn the neuron on, and we'll vasodilate when we turn the same neuron off. So the sympathetic controls our blood vessel diameter, not the parasympathetic. Now, why is it so important to change or be able to regulate blood vessel diameter or radius? It's because, again, it has a fourth power influence on resistance. So it has a huge influence. And so when we need to redirect blood flow, this is how we would do it. We would vasoconstrict areas where we would want less blood to go, and we would vasodilate the areas where we want more blood to go. So our heart's job is just to contract and create the pressure to send blood out into our blood vessels. But wherever we have less resistance, meaning wherever we're vasodilated, that's where more blood will flow. And wherever we're vasoconstricted and have more resistance, that's where less blood will flow. So so we talked about arteries, and then we talked about arterioles, where we constrict and dilate. So now let's move on to capillaries. So our capillaries are important because this is where your exchange takes place. So you are going to exchange things like glucose, oxygen, carbon dioxide, protein. So all of the things that are going to move in and out of our blood moves through capillaries. And it can move via paracellular transport, which is between endothelial cells, or transendothelial transport, where it moves actually through the cells, so it has to go through the cell wall. Small dissolved solutes and gases can just move by diffusion, which we know is determined by, the movement is determined by the concentration gradient. And then larger solutes and proteins might need help through vesicles. So that would be our vesicular transport, where we can do things like transcytosis. That's where we endocytose on one side, take the entire vesicle to the other side, and exocytose. So a lot of this was covered in some of the earlier chapters. Now, fluid exchange at the capillaries is determined by two major pressures. The first thing I want to mention is here's our arterial side of the circulation, here's our venous side, and essentially this is the capillary that's in between. The first thing to note is we will always filter first, and then we will absorb. So we're going to filter out fluids and electrolytes and ions and solutes, and then we're going to absorb them back, or most of them back. We really don't want excess fluid in the lymphatic or in the interstitial space, because then we'll suffer from edema. If we ever have more fluid just kind of hanging out in the interstitial space, then the lymphatic vessels will pick it up. And that's where the lymphatic system comes in. So it'll pick up the excess fluid, and then eventually it'll drain it back into our circulation. But the two pressures that influence filtration and absorption are hydrostatic pressure and osmotic pressure. So hydrostatic pressure, wherever you have high hydrostatic pressure, it's going to push fluid out from that area. So if we have a lot of hydrostatic pressure in the blood vessel, that hydrostatic pressure is going to promote filtration. It's going to promote the movement of fluids out of the blood vessels into the interstitial space. Colloid osmotic pressure, wherever you have that, that's going to promote fluid coming into that area. So if we have a lot of colloid osmotic pressure in the blood vessel, that's going to promote absorption into the blood vessel. And these two pressures will determine how much we filter and how much we absorb. This will come up again when we get to the renal lecture. Okay, so again, hydrostatic and osmotic pressures help regulate the flow, the bulk flow, which is the mass movement of fluid between the blood and the interstitial space. And filtration is the fluid movement out of the capillaries, and that's really going to be promoted by the hydrostatic pressure. And absorption is the fluid movement back into the capillaries. And the osmotic pressure will really help with that. And like I said, the lymphatic system plays a really big role here. It will return any excess fluids and proteins. It'll pick it up and return it to the circulatory system. But the lymphatic system will come up a couple more times. We'll talk about it in the digestive lecture because it's also involved in fat absorption. So when we eat fat, and bile helps us emulsify it, and then when we absorb it, instead of going directly into the capillaries, it goes to the lymphatic vessels first, and then it will get transferred into the circulatory system. And then the lymphatic system also obviously is involved in white blood cell function. So it helps filter a lot of pathogens. We have our lymph nodes that have a lot of white blood cells ready and waiting to deal with any pathogens it comes across. So I mentioned edema briefly, but it's when you have swelling due to accumulation of fluid in the interstitial space. So if you filter more fluid than you absorb, that fluid is going to stick around in the interstitial space, and that can lead to edema. Again, the lymphatic system helps prevent that, but you can overload the system. And so if the fluid just kind of builds up, we have that swelling happening. Now let's take a look at blood pressure and blood pressure control. So we know the cardiovascular system can influence blood pressure. And we also know that the medulla is the cardiovascular control center. So when we need to increase blood pressure, we would turn on the sympathetic output. When we need to decrease blood pressure, we would turn on the parasympathetic output. So let's take a look at what these nervous systems will do in terms of the cardiovascular function. So let's start with the sympathetic. So when we need to raise blood pressure, because our baroreceptor sensed that blood pressure was low, the medulla will turn on sympathetic output. The sympathetic neurons, and we're going to follow the arrow, will directly influence the SA node. So the SA node will beat autorhythmically, but the sympathetic neuron will just speed it up. So we'll increase heart rate. And we know when we increase heart rate, we're going to raise blood pressure. The sympathetic neurons also influence the muscles, the ventricles, and that's because it can cause increased contractility. So basically, it makes your heart muscle contract with more force. And if your heart is contracting with more force, you are increasing stroke volume, and we know when you increase stroke volume, you increase cardiac output, and that raises blood pressure. The sympathetic neurons are also going to our arterioles and our veins. And remember, both of these are capable of constricting. So the sympathetic output is going to cause vasoconstriction of our arterioles, and that will raise our blood pressure. Now, what if blood pressure is too high? Then we will turn on our parasympathetic output and essentially turn off the sympathetic. Now let's take a look at the parasympathetic neurons. Notice there's only one arrow coming out of here, and it only goes to the SA node. So the parasympathetic neuron will influence the SA node and tell it to beat slower. So lower heart rate means lower cardiac output and less blood pressure. But notice there's no influence directly to the ventricles or to the arterioles. So why will your heart beat weaker, and why will you vasodilate when you activate the parasympathetic? It's because you are turning off the sympathetic. So since the sympathetic is not telling the ventricles to contract with more force, they will contract with less force, and contractility will reduce. Because the sympathetic is not telling the arterioles to vasoconstrict, they will vasodilate. Remember, we have tonic control of our blood vessels through the sympathetic neurons. So all of this is shown here. So again, let's say we have a rise in blood pressure. We are going to then turn on the parasympathetic output and turn off the sympathetic. By turning off the parasympathetic, the SA node will slow down. But by turning off the sympathetic, the blood vessels will vasodilate and the force of contractions will reduce. On the other hand, if we have a drop in blood pressure, for example, with orthostatic hypotension, this is the drop in blood pressure that happens when you stand up too quickly, we want to raise blood pressure. So the sympathetic nervous system will be turned on, parasympathetic will be turned off. And the sympathetic will actually influence all three. It'll increase heart rate, increase contractility, and vasoconstrict. All right, so we are going to end with cardiovascular disease. And cardiovascular disease is important to understand because it's a major issue in the United States population. So there are several risk factors that increase your risk, and some are controllable, some are not. So some of the risk factors that you cannot control would be things like your gender. So men have a higher risk than women. Age plays a role. So as you get older, your risk increases, as well as family history. The good news is there are controllable risk factors. So smoking is a risk factor, so avoid smoking. Obesity is a risk factor, so make sure you are able to maintain your weight and lose excess weight. And living a sedentary lifestyle is a risk factor as well. So this is not exercising. So begin an exercise regimen. And then untreated hypertension. So high blood pressure that's not treated is a risk factor as well. Other risk factors include increased blood lipids, as well as diabetes mellitus. So blood lipids can lead to, for example, atherosclerosis. Now, atherosclerosis is hardening and loss of elasticity of your arteries because of fatty deposits in the blood vessel wall. And so when you lose that elasticity in the arteries, then that's going to really increased blood pressure, and your cardiovascular system will have to just work harder. Your heart will have to work harder, and your body will kind of suffer because less blood may be getting to you to your system. And then plus that can lead to left-sided hypertrophy and left-sided heart failure. So there's a whole snowball of things that can happen. Now, there are different types of lipids. We have HDL and LDL. HDL stands for high density lipoprotein cholesterol. These are denser in denser in protein and less dense in lipids. So these are the ones that are more desirable, and you actually will reduce your risk of cardiovascular disease by having high levels of the HDL. LDL is the one that you don't want. So the low density lipoprotein cholesterol has a lower density in protein and a higher density of lipids. And so this is really associated with heart disease. And then diabetes mellitus, which we know is the inability to maintain your blood sugar, also contributes to atherosclerosis. And we mentioned how untreated hypertension is a risk factor. So hypertension is high blood pressure. Our carotid and aortic baroreceptors, which sense changes in pressure, can actually adapt. So when your blood pressure is always high, your baroreceptors can adapt to that and kind of decrease the response because it's just always high. Hypertension is a risk factor for atherosclerosis and cardiovascular disease, and it can lead to hypertrophy of your heart, especially your left ventricle, if you have high blood pressure in the systemic circuit. And treatments, we can use calcium channel blockers. So we just learned how calcium is important for smooth muscle contraction, especially extracellular calcium. So if we block calcium channels, it can reduce the amount of calcium coming into the smooth muscle, causing them to not contract. So we'll vasodilate as opposed to vasoconstrict. Diuretics will help because we're going to be able to eliminate more fluids in the kidney. So less fluid in the blood means less blood volume, which lowers blood pressure. Beta blocking agents help. Well, beta blockers are blocking the sympathetic nervous system because remember, these are beta adrenergic receptors, and that's part of the sympathetic. And we know sympathetic raises blood pressure, so blocking the sympathetic receptor will lower blood pressure. And then we have ACE inhibitors. So ACE is part of the RAA system, renin, angiotensin, aldosterone. Renin is released by the kidney, and the end result really is it raises blood pressure. And ACE is part of the action of renin. And so if we block ACE, we're essentially blocking what renin will do to our blood pressure. As a result, blood pressure will not be elevated. It'll help reduce it. Essential hypertension is when there's no clear cause other than just you have high blood pressure, maybe family history played a role, versus secondary hypertension, there's like an identifiable cause. So let's say you have less blood going to the kidneys because of some obstruction or blockage, and so your kidneys are releasing a lot of renin, and that's why you have high blood pressure. Then you can fix that by trying to restore blood flow to the kidney. So those are known as secondary hypertension. And that's it for this chapter. Thank you.